everyone, and welcome to the session HPC Power and Breakthroughs in Life Sciences. It's really a pleasure for me to chair this session. We have two speakers, Dr. Natalia Jimenez from Atos, who is our life science industry lead, distinguished expert and member of the scientific community. She's going to give us an overview of the many areas in life sciences that could benefit from the acceleration provided by high-performance computing. Then she will hand over Dr. del Castillo, Scientific Director of the Translational Genomics Core of Hospital Ramon y Cajal in Madrid, who's going to tell us about the positive impact of HPC in the diagnosis of rare disease. I take the opportunity to thank him for joining us today. And now I leave the floor to Natalia.
Hello, I'm delighted to be here today to tell you about the many use cases of high-performance computing in the life sciences industry. But before doing so, I would like to frame high-performance computing in the whole Atos value proposition in life sciences. As you can see in this slide, this is a very simplistic illustration of a workflow where you have a sample that you put in a sequencing device, could be a sequencing device or any other kind of high throughput device. The result or the output you get from this is a big data. In this case, a whole genome sequence that needs to be transformed into actionable insights. In this setting, which is a clinical setting, this would be used for diagnosis or for prescribing personalized treatments. So in all this transformation, we are finding different challenges that we need to face. And the challenges could be related to the data acquisition and validation, or also to the data processing, correlation, exploitation, and even with the data integration. The Atos value proposition is to design solutions, end-to-end solutions to face all those challenges by applying different technologies. Here, we are going to focus exclusively in the high-performance computing capabilities to accelerate the processing of raw data. Let's go to the first use case. This is on structural biology. Structural biology is an area of life sciences that studies the structure of macromolecules. It can be used using different high-throughput devices to do that, but we are going to focus in the electron microscope. Basically, you go to the microscope, you take your images. The images are big data, and the reason is because these images could be equivalent to 6.5 millions of pictures you can take with your smartphone. So you need to process all this data to get the 3D model. And the 3D model is key because from the understanding of this structure, you can get the idea of what is the function of the macromolecule you are studying. So in this processing, high-performance computing plays a very important role because it accelerates all this processing. So in this specific example is with the National Center for Biotechnology, which belongs to the Spanish Research Council. And they are not only producing these 3D models, but also they have in place a platform for electron microscopy, and they are delivering tools and services through this platform. So we are helping them to be more efficient in what they do. And one important thing is that they recently solved the 3D structure of the spike of a SARS-CoV-2. This is precisely the protein that the virus uses to enter into the cell. So very important achievement. Now we are moving to this institute of wine and grape wine sciences, and this is co-located with a Spanish qualified designation of origin in the area of North Spain. This institute uses high-performance computing for genomics, for genomics, because they are trying to identify what are the variants that could be used to improve the organoleptic properties of wine. So we can say that we are in some way helping them to produce better wines. Now we are moving to the west from North Spain to the UK with the Palbret Institute. This institute, they are working or they are studying viral diseases in poultry and livestock. One important thing is that they are also studying zoonosis, which is the process by means the viruses are jumping from animals to humans. And this is what happened with SARS-CoV-2. So they are using heterogeneous supercomputing capabilities to sequence viral genomes and also to get more understanding about the interactions between host and pathogens, and also to model the spread of the diseases. We have been working for more than a decade now with the National Center for Genomic Analysis. This is the second largest center in Europe in terms of sequencing capabilities, after the Sanger Institute in the UK. They have the capability, because they home like 12 sequencing devices, to sequence 8 whole human genome sequences per day. So they are delivering human genome services to hospitals, research centers, healthcare systems, pharmaceutical companies. But apart from doing so, they are also working in very interesting or relevant projects, such as, for example, the sequencing of the albino gorilla or the Iberian lynx. One important thing is that they've identified the variants that are making of the albinism. And for the second one, for the Iberian lynx, as you know, this is an endangered species. So they realized that one of the main reasons why that's happening is because this lynx is very prone to suffer infections. In other words, his or its immune system is faulty. But apart from gorilla or lynx, they are also sequencing agricultural useful species, such as, for example, tomato, melon, or olive tree. And the reasons to do that is because they can improve the crop production, for example, by making those species resistant to drought or resistant to plagues. Well, one of the main areas in which Atos is working is in the enablement of precision medicine. What we try to do is to transform all this big data that is being generated in the clinical environment into something that you can use to implement use cases. Basically, transforming the fresh and bone human being into the digital human. So as you know, the human body is a big data producer, so it's producing data by omics technologies, by different modalities of image data, metadata, wearable devices, sensors. So you need a lot of different technologies in order to process the information, to correlate the information, and to transform that information into useful insights. So this is all about precision medicine, but Dr. Del Castillo will tell us more about that. Just to finish, I would like to mention that all these use cases are possible because Atos is the #1 in Europe, South America, and India in high-performance computing. We have more than 400 customers in high HPC and more than 700 experts. And as we realize how powerful we are in managing high-performance computing, we decided to focus our efforts in life sciences industry. For that reason, we created the Life Sciences Center of Excellence, which is an exploration center to bring technologies closer to research and to the clinical environment. That's all on my side. Now I hand over Dr. Del Castillo, who is going to tell us about HPC.
Thank you, Natalia. Thank you very much. I'm very happy to be able to present our work here in this excellent venue. And I'm going to talk about the help that HPC is providing to research in genomics. And my first point would be to make you understand what is precision medicine. And precision medicine is a relatively new approach. Over the years, people working in the medicine have realized that there are no diseases. They are just patients with different manifestations of the same disease. And these differences come because the patients have among them genetic, environmental, and lifestyle differences that make that treatments and diagnosis are not the same from one individual to another. So we have now, over the last 15 years, switching our focus from treating a disease to treating individual patients. And the crux here is that most of the meaningful data that we get now on differences are coming from genetic analysis. And this analysis have a problem that we have to generate data and analyze it quickly, securely, cheaply, and we have to enable continuous reanalysis because science progresses, more and more data are available, and things that would have been overlooked previously needed to be reanalyzed in the present moment. The second problem here is also that actionable information must be recognized and made available to the people that have to take decisions as soon as possible. And finally, we have an issue with the security of data. Data has to be stored securely and retrieved efficiently and kept under the constraints of the GDPR, the data protection systems in every part of the world. So now I would like to give you an idea about the nature of the challenge we are facing daily. So if you go to the Guinness World Book of Records, what you will find is that the longest book known is À la recherche du temps perdu de Marcel Proust. It's about 10 million characters in length. So what we would do if it was just the human genome to do the analysis of this book is just shred it into just pick up 50 copies of the book, shred them into small fragments, just 150 character long, and then we have to reassemble all the novels quickly and efficiently. Then we have to note all the differences and typos among the different 50 copies and compare this with the original edition. Then we have to tell which typos are changing the meaning and which are not. And then we have to quickly write an actionable report on those differences. So you see that this is clearly a job for HPC. But if you are thinking that just this is going to be a challenge, now we have to put this in the scale of the human genome. Because the human genome is about 300 times as long as a single copy of À la recherche du temps perdu. And it is written, instead of a 25-letter alphabet, it is written with a 4-letter alphabet, which is making this task much more difficult. Also because the genome contains very similar repeats in which subtle changes are meaningful. And to boot, you must be quick in reassembling the data and analyzing it because this is not a theoretical exercise. The genome that you're analyzing is belonging to a fellow human being that is suffering, maybe in critical condition, and your analysis is the key to a successful treatment. So you see that really it's a true challenge. And what is the scale of the challenge as it stands now? Well, a few years ago, the problem might have been generation of the data. But now, generating data for a human genome is both affordable, it's about $1,000, and it is quick. We can do this within the scale of days. But unfortunately, the analysis time as it stands today is taking weeks. So we have to do a certain job here because the bottleneck is getting enough HPC power. What are we using all this power for? Well, we are analyzing rare diseases. Rare diseases are altogether very common. About 1 in 20 Europeans has a rare disease, but individually, every rare disease is very, very uncommon. About 1 in 2,000 to 1 in 10,000, or maybe some diseases, maybe just 50 people with the disease all over the world. We are focusing on lysosomal disorders. Those are a kind of metabolic disorders, and many of them can be treated. And we are really putting a lot of effort in speeding up a diagnosis, because for those diseases that can be treated, it is critical to give the treatment early. So you can see in the slide somebody that is in a wheelchair. This is a child with pump disease, one of the diseases in this group. And now, for example, in Taiwan, people that may be at risk of this disease are very quickly diagnosed within the first 6 hours after being born. And if we confirm that they have this disease, treatment is applied. And then in a few months' time, you have a child that is able to run normally and it is not confined to wheelchair. So you see how critical it is to have the disease properly diagnosed, all the variants found, all the actions taken and treatment applied in time to avoid a negative outcome for the disease. But we are not only doing this. Genomics is a powerful tool, and we are tackling even COVID-19. So we are currently within my platform studying 2 different projects about COVID-19. One is related to what the virus is doing. We are studying SARS-CoV-2 samples from affected individuals, because what we want to check is whether the pathogen is evolving, and we can identify mutations that may help it become more infective or less infective, whether it's providing resistance to some of the things that we can use to treat it, and so on. And most importantly, from my point of view, we are now leading a second effort to identify the host genetic differences, the differences among the patients that are underlying a worse prognosis of COVID-19. This is not so important for the general population, but you know that with this disease, the problem is that it is flooding the resources of the health systems. So if we are able to tell which of our colleagues that are treating patients are at risk of developing a very severe disease, we can just pull these individuals away from the front line, and like this allocate resources more efficiently to fight against the virus. And with this, I am finished, and I would just pass the floor to Albert to lead the question and answer part.
Okay, thanks to Natalia and Francisco for your presentations. We received some interesting questions for you here, and we'll now take time to answer this. First one, I think it's for Natalia. Natalia, you mentioned a center of excellence. Where it will be located and what type of activities will do?
Oh, that's good that I can tell about the Center of Excellence. It would be located in Cambridge, UK, but the scope is global, is worldwide. But we decided to have the place here because it is where all the life sciences hub is located. The kind of activities we are going to do are related with the exploration of new technologies. We are going to, as I said at the beginning, to bring technologies closer to our customers. The idea is to explore through a proof of concept, proof of values, innovation workshops, demos. We are also thinking of having PhD students with us. So sky is the limit for this kind of activities.
Okay, thank you. A new one for Dr. del Castillo. Is the same method valid for any rare disease?
Well, in principle, yes, but also I would say no. Particular diseases have particular problems and may require some specific answers, but the whole framework, there is a whole body of approaches, of computational and bioinformatic approaches to tackle most of the things. Just a few differences may require specific programs or computational approaches, but most of the time it would work for any kind of rare disease.
Okay, thanks. Now a good one, probably for both of you. How much investment in HPC is needed for genome analysis?
Wow, that's a good question.
I can take this. Yeah, I think there is not a right answer to that, but a range of different options. It will depend. So if you are a genomic production center, obviously you have to spend a lot of money in high performance computing because your whole business is all around delivering services around genomics. But if you are a hospital, for example, in the case of Dr. Del Castillo, there is a difference because you are only using, well, you are using genomics for diagnostic purposes, but it's a more focused kind of activity. So I will say that the methodology we have in place in Atos is through our reference architecture. So we have a way by questioning the customers about the projects, for example, number of genomes that they need to produce, or what are the data retention times, or many other questions, we can figure out what is the right architecture for their purposes. So it's a very customized approach. There is not, there is not a right answer to that.
Well, on my part, I would like to point that this is a very novel field, and hospitals, the management part, is not yet ready to invest enough in this technology. So, for example, for research purposes only, we have a cluster in our hospital that was set up by Atos, and it cost us something like 250,000 euros. And this is allowing us to produce whole genome data analyzed in about a couple of weeks. So this is really not good enough for a clinical setting. What you have to do is keep in mind that the market will balloon somewhere in some time. And whenever you are setting any kind of architecture, make it modular so that you can scale it up easily to meet the challenges. I think that now bioinformatics has been put in very much in the limelight because of the need for bioinformatics because of COVID-19. So this is really, from a market point of view, an excellent time to start pushing for most important investments in bioinformatics in the health systems.
Okay, thanks. New question for Dr. Del Castillo. Is there any preliminary outcome from your COVID projects?
Unfortunately, no. We have now started sequencing 300 samples from patients, and both genomics data from patients and from the viruses that were invading them. And we have not yet any preliminary data. We have to collect all the data and analyze all together. So I cannot really give you any ideas more than it has already been published by other centers.
Okay. Then thank you all for being in our live session about HPC in life sciences. The recording will remain online for you to look at it back. And we hope it has been of your interest, and see you soon.
Thank you.
Thank you.